All right, hello and welcome back. Today we are going to go into topic 3 in the Center High Science 10 biology booklet. We're going to talk about the cell membrane and transport systems. The goal is to explain the difference between passive transport mechanisms and active transport mechanisms, define things like osmosis, differentiate between things like facilitated diffusion and normal diffusion, and describe the surface area to volume ratio of cells. That's a lot of information. We're going to go through it all today. Every living cell must obtain nutrients. That means that they must take in nutrients or food from its surroundings, and every living cell also must release waste. I zoomed in on the screen more than I meant to, so that it can get rid of the substances that it doesn't want. Here's a cell. This cell is going to take in some nutrients. Carbohydrates are an example of a nutrient. That's another word for like sugars, simple sugars, complex sugars. These are going to be an energy source. Oxygen is going to be required to extract energy from that energy source through cellular respiration. So both oxygen and carbohydrates are our nutrients. And carbon dioxide is something that we're going to produce as a byproduct of cellular respiration that we don't want. So that will be a waste. Because you have these nutrients and because you have these wastes, you need to come up with, or cells need to come up with a method of moving those molecules. Molecules are moved or are transported across a cell's membrane. 2 lessons ago, we talked about the structure of the cell membrane, and we said that it's a phospholipid bilayer. I'm just going to highlight the parts of this word here, phospho and then lipid. Red and blue is what I'll stick with. Red because it's red here, and then blue for lipids on this side. A lipid is another word for a fat or an oil. And the main thing that I want you to know about lipids is that lipids do not mix with water. The phospho part refers to the phosphate group, which is written as PO43-. If you have your Science 10 periodic table and you look at the table of common polyatomic ions that we looked at so much during the chemistry unit, you may remember phosphate as PO43-. That's attached to the lipid. The phosphate part likes water. So here's a little diagram of our phospholipid. Here's our phosphate. I'm just going to write on it PO43-. And this part likes water. We say that it's hydrophilic. Hydro meaning water, and philic meaning to love. This part on the bottom, which we have 2 of those, these are our 2 lipid tails. 2 lipid tails. The 2 lipid tails do not like water. We describe our lipid tails as hydrophobic, meaning they are afraid of water. They don't want to be part of or close to the water. Because we have this sort of nature where one end of the phospholipid wants to touch water and the other end of the phospholipid doesn't want to touch water, the phospholipids arrange themselves into a bilayer. And the cell membrane is an example of this bilayer. You see that you have on the inside here all of our lipid tails. On the inside of the phospholipid bilayer, we have our hydrophobic lipid tails. And they arrange themselves towards the inside so that they don't touch the water. And then all along the top here are our phosphate heads, which are hydrophilic. I know that the diagram below has these exact same labels, but if you write it out, then it will help you remember it. The hydrophilic end points towards the water, and the hydrophobic end points towards the center. A bilayer just means that there's two phospholipids, two layers. Bi means two. Layer on top with the tails pointing in, and layer on the bottom also with the tails pointing in. Water on either side. One of these sides will be the external environment, and the other side will be the cytoplasm. So if we look at this diagram here, arbitrarily, I'm just going to choose that the bottom side is the water of the cytoplasm, and the water at the top side is our external environment. And our phospholipid bilayer is going to separate those two. The cell membrane isn't just phospholipids, though. It also has protein molecules mixed in with the phospholipids. And each of these proteins has their own function. Remember when we were talking about the organelles, that ribosomes make proteins, and proteins are these functional pieces inside of the cell. Each of these will have a different job. Here's 4 examples of proteins inside of the cell membrane. You can have channel proteins. Channel proteins allow particular molecules or ions to cross the cell membrane. Think of a channel protein as just an opening for things to move. Now, a channel protein can be specific to a specific type of molecule. Often it is. It says here that it allows particular molecules or ions. Above the word particular, I'm going to write specific. That means that you can have a channel that is going to allow molecule A through, but it won't allow molecule B through. And this will be useful, as we'll see later on. Similar to channel proteins, we have carrier proteins. Carrier proteins also allow a specific molecule or ion to cross the cell membrane, but instead of just being an open channel, there is an interaction here. So you'll see here that the carrier protein is going to grab on to whatever this solute is that is moving across the membrane and push it out on the other side. In this particular diagram, we'll just say that the outside here, this is the external water. And the inside here is our cytoplasm. And so we have this carrier protein that very helpfully will move whatever this solute is. We're going to say it's something that we want, maybe sodium ions, across into the cell. We also have receptor proteins. That's supposed to be an E as our second letter here. Hopefully you're going to notice a theme here, that each receptor protein has a particular shape that allows a specific molecule, like a hormone, to bind or attach to it. If you take a look at this particular diagram of a receptor protein, this blue rectangle would fit in the receptor, but the other molecules would not. So the receptor protein is specific to a molecule. It is specific to the blue molecule. And finally, we have enzymatic proteins. Enzymatic proteins. These are going to facilitate chemical reactions. What this is trying to show you is that we have some molecule, this substrate, that binds with the enzyme, and then a reaction occurs and it's transformed into a different molecule. Receptor proteins here often are used to allow cells to communicate with each other, and enzymatic proteins are going to carry out a specific reaction. Maybe it's related to generating energy or moving substances around. But here's 4 general categories of proteins that you can find in the cell membrane. Before we move on to the next page here, I want to take a look at this diagram at the top. Sometimes we call this diagram the fluid mosaic model. Fluid mosaic model. A mosaic is an image that's made up by a bunch of smaller pieces. Often you see it as like a public mural or an art piece where you have a lot of small little tiles or glass pieces that are put together to form an overall image. A cell membrane is not just one thing. It's not just phospholipids. You have all of these different things attached and embedded in the membrane. And it's fluid because it's flexible. It can move around and things can move across it. We've talked about proteins. These are these orange and blue, or I guess kind of greenish pieces. These are all proteins. One piece that we haven't really talked about that I do want to focus on here is cholesterol. Cholesterol is part of our fluid mosaic. And I'm just going to highlight a couple pieces of cholesterol inside this membrane here. It's a type of lipid that helps to stabilize the membrane, especially against temperature changes. And that might be something that's important to know. Cholesterol is a type of lipid that helps to stabilize the membrane especially against temperature changes. So as you warm up molecules, they move faster. And if you want to hold this membrane together, the cholesterol will help to stabilize the membrane as the temperature increases or decreases so it doesn't fall apart. I have a bit of a animation. Some brilliant person made on Google Slides. It wasn't me, even though that sounds self-serving. I'm just going to take my face off the screen. There we go. This will bring us through some types of membrane proteins. Channel proteins. These are going to transport molecules past the cell membrane like a tunnel. So if this is our outside the cell area, here's our inside the cell or our cytoplasm, extracellular. Here's a bunch of different materials that can pass through the channel protein that wouldn't be able to pass through the membrane. Carrier protein. This will transport a very specific molecule past the cell membrane, like a tunnel with a door, and only specific molecules have the key. So unlike the channel protein, which the size allowed all sorts of molecules to pass through, this one only allows the circle molecules to pass through. Now, this animation makes it look like the channel protein can't be specific to molecules. There are channel proteins that can be specific to certain molecules, especially based on size, but this carrier protein interacts with the circle molecule. That's what makes it particularly different. A receptor protein will bind a specific molecule and then send a signal. The square molecule here is able to fit into this receptor molecule, which then it will cause some sort of change, and this receptor protein can signal something within the cell to take place. Lots of hormones will bind to receptor proteins to cause changes within other cells. And then there's an enzymatic protein. It will bind a specific molecule, just like the receptor protein was, except it's going to cause a chemical reaction or speed up a chemical reaction, which could be lots of different things. It could change the shape of a protein, It could combine two molecules together, or it could break a molecule apart. So in this example, we have a square molecule enter, and then we change it into a circle molecule. And the enzyme makes this reaction happen. Like the receptor protein, it binds a very specific molecule, but unlike the receptor protein, instead of sending a signal, this molecule is going to cause a reaction to happen or facilitate a reaction. Okay, to the next page. Here's another diagram that shows our 4 types of proteins. Here we have a channel because you can see the molecules moving from one side into the cell. This is a carrier because it doesn't just move through an open hole. It has to interact with the protein. Here we have a receptor. And here we have an enzymatic. You can see that one molecule enters, and then a different molecule, a different product, is what leaves on the other side. All right, that's what the membrane is made of, phospholipids and proteins and cholesterol. But what does it do? Cell membranes are semi-permeable, which means it allows some things to pass but not others, like a screen door. So if it's hot in the summer, you want to let air into your house, but you don't want to let the bugs in. You leave the screen door closed, but you open your main door so that air can come in to cool down your house, but the bugs don't fly in. That's semi-permeable. It allows things like air to pass through, but it doesn't allow animals to come in. Similarly, the cell membrane allows oxygen and nutrients into the cell. It keeps out larger molecules. It also keeps in larger molecules. Because it would be a shame if you lost all of your organelles from inside your cell. All of these are trapped inside, and then some nutrients are allowed to move in. Cell membranes allow food, water, and oxygen gas in for animals, and then remove wastes and waste gases like carbon dioxide. Movement is regulated based on size. So some invaders, like bacteria and viruses, are too big to be allowed in. There are some large molecules that you want. Large molecules that you want, so large nutrients needed by the cell, such as sugar, are transported by proteins into the cell. They cannot pass through the membrane, so they need a doorway. They cannot pass through the membrane, so they need a doorway of sorts. The cell membrane is the organelle responsible for transportation of materials in and out of the cell. Before we talk about the methods of transportation, it's going to be important to discuss what we call the particle model of matter, because we're looking at the movement of particles. First of all matter is made of particles. Particles can be atoms, ions, molecules. The particles in different substances may be different in size and shape. I'll just make a little note here. Examples of particles. These could be atoms, ions, molecules. These are particles. All matter is made of particles. The particles of matter are constantly moving. They're in motion. Particles move slower in solids and faster in gases. Adding or taking away heat will affect the movement of particles. The particles of matter are attracted to one another or are bonded together. Particles have space between them, and those spaces are, we'll just correct this word to spaces. Particles have spaces between them that are smaller in solids, except for ice, which strangely has greater spaces than the liquid form water, and greater in gases. These spaces may be occupied by the particles of other substances. So, for example, if you have liquid water, the spaces between those water molecules can be occupied by things dissolved in the water, or solutes. Here's some pictures. This is our gas state. These are our different particles. They are far apart from each other, and they are moving very fast. This is our liquid state, where the particles are closer together and moving not as fast. And then this is the solid state, where the particles are as close together as they can be and moving slower, probably just vibrating in space rather than moving around the container. Passive transport. Passive transport uses no energy. There is no energy required. Remember that ATP is our usable form of energy. Back when we were talking about the different cell organelles, we said that ATP is made in cellular respiration, and it is one of the usable forms of cell energy. Every time your cell wants to do anything, it needs to spend ATP. Think of it like money for the cell. There are three kinds of passive transport. This is transport that requires no cell energy: diffusion, osmosis, and facilitated diffusion. Passive transport relies on something called a concentration gradient. A concentration gradient is a difference in concentration between two areas. Before we can understand a concentration gradient, we should define the word concentration. In this context, concentration doesn't mean focus. We're not saying like you're concentrating on studying or you have great concentration because you can do math problems for a long time. Concentration here refers to how much of a substance is in a given area. Take a look at this diagram here. We're going to pretend that this is the same section of membrane in 3 different time periods. So this one here is going to be at time 1. This is at time 2. And this is at time 3. At the start, during time 1, there are no hexagons in the cytoplasm. And there are lots of them, lots of these particles in the extracellular fluid. Extracellular means outside the cell. This is going to be a high concentration. I'm actually going to use red for high concentration. This here is a high concentration. And then on the other side, on the inside here, this is a low concentration. Because we don't have any of these particles. You can't get lower than having none of them there. Note that we move these hexagons from the area of high concentration over time to the area of low concentration until the concentrations are equal. This is movement along a concentration gradient. So the particles, these hexagons, move from high concentration to low concentration until concentrations on either side of the membrane are equal. At the end here, both of these have the same concentration. At the start, we had a higher concentration outside of the membrane, and at the end, the concentrations are equal. So they must have moved from the high concentration to the low concentration. Diffusion is the description of this process. Diffusion is the movement of molecules from areas of high concentration to areas of low concentration. This is because of the constant movement of particles. Particles are always moving around, so eventually you're going to end up with particles evenly spread out. Diffusion eventually results in something called an equilibrium. So if you look at this diagram here, we place a solute inside of our solvent, and then eventually the solute particles, which are more concentrated at the top of this beaker, move around until they are evenly spread out. This is the beaker at equilibrium. At equilibrium, the concentration of particles is evenly distributed. You have the same amount throughout the volume. Now, the particles do not stop moving at equilibrium. They are still moving around back and forth. It's just that the movement is back and forth at an equal rate. So again, here's another diagram. We have high concentration on this side. So I'm just going to write high concentration on this side. We have a low concentration on this side. Particles move from high to low until you've reached equilibrium. But even at equilibrium, they're still moving around. You'll still have particles move to this side from that side, and particles move from this side to that side. It's not that they stop moving, it's just that the rate of them crossing is equal. So the overall concentration remains the same. Here's a diagram here. We've got a bunch of particles in the extracellular fluid. So I'll write out here that this is my extracellular fluid. Here is my high concentration of particles. And then these particles are going to diffuse across the membrane to an area of low concentration. And this movement into the cytoplasm is diffusion. If you want to label more things, this here is a phospholipid with a phosphate head and lipid tails. That's diffusion. Diffusion, it moves across the membrane on its own. Facilitated diffusion involves channel proteins and carrier proteins. You need to have channel proteins and carrier proteins. And it requires no energy input. No energy, which remember is our ATP from the cell. All it's doing is you are allowing a pathway for particles to diffuse across the cell. It's still diffusion. We're moving from an area of high concentration. These square brackets here, the square brackets here mean concentration. So on the outside of the cell here, we have a high concentration of these orange dots and a high concentration of these purple triangles. But then you need channel proteins and carrier proteins to allow things to cross. This channel protein here is specific for the orange dots. It's specific for this solute. And this carrier protein is specific for the purple triangles for that solute. So I'm just going to write here, this is the same as diffusion, where you go from high concentration, I won't use the square brackets, I'll write the words, where you go from high concentration to low concentration. The only difference is that a protein provides the pathway. All right. I'm going to pull up another simulation here to help describe this. Let's see if I can zoom in on this a little bit. Okay. Here's my membrane. I'm going to say that where my mouse is on the top is outside my cell, and on the bottom is the inside of my cell. I'm going to add some particles. Oh, wow. This is not working the way it's supposed to. Give me a second. Let's try this one more time. There we go. I'm going to add a bunch of particles to the outside of my cell. The concentration outside my cell is much higher than inside my cell. This graph shows the concentration. And then we're going to add a bunch of blue particles to the cytoplasm, the inside of my cell. These particles are too large to diffuse across the membrane, so they need a protein. If I add a protein that allows green particles to cross, then green particles will start to diffuse into the cell. If you're watching the green particles, you'll note that some of them are actually going to start to leave the cell. That's because they can move both ways. But overall, the concentrations will start to even out because more particles will leave the area of high concentration and go to the area of low concentration. I can speed this up so that we can reach equilibrium a little bit faster. Now, if you're comparing the size of these green columns, the concentrations of the green particles are equal on either side of the membrane, roughly. It keeps changing, though, because particles can keep moving back and forth. So it's in a state of equilibrium, but that equilibrium is dynamic. It keeps moving. If I add some proteins to allow the blue molecules to move across, they will diffuse from the area of higher concentration to the area of lower concentration until those concentrations are equal. This simulation also has these gated channels. Some proteins can open and close to help change the concentrations, but that's a little bit beyond Science 10. What I want you to see here is that we have reached a state of equilibrium where the concentrations of both solutes are roughly equal on either side of the cell membrane. The last type of passive transport is called osmosis. Before we talk about osmosis, we need to talk about a couple key terms. First is solute. Solute is the thing that dissolves. So the solute is the thing that dissolves. For our examples, it's going to be mainly sugar, salt, ions, proteins, that sort of thing. Those are our solutes. The solvent is the liquid in which the solute is dissolved to form a solution. The solution is the mixture of the solute and the solvent. The solvent for our cells and the cells of all living things is water. Our solvent is going to be water. Osmosis is similar to diffusion, but it involves only water. So you can think of it as the diffusion of water across the membrane. Water molecules move from their area of high concentration to their area of low concentration. Take a look at this beaker here. This beaker here has a selectively permeable membrane. It does not allow the sugar molecules to cross. Sugar cannot cross the membrane. Instead, water will cross the membrane. Sugar cannot cross, water can. For our previous examples of diffusion, we saw the solute move from the area of higher concentration to the area of lower concentration. If the solute concentration is high, that means the solvent concentration is low. We have a concentrated sugar solution, which means that we have a lower water concentration. And so the water will move from the area of a high water concentration to a low water concentration. This is osmosis taking place. So you would see that the volume in this side of the beaker increases as water moves to try and equalize these concentrations. Once again, diffusion. Solute molecules move. The solute molecules move from a high concentration to a low concentration. Here, the solute is in a highly concentrated bundle, and then it's going to move to spread out throughout the entire beaker. If the solute cannot move because of a semi-permeable membrane, then the solvent will move. Osmosis requires the membrane. This semi-permeable membrane is required to prevent solute from moving. So in this particular side of the beaker, if we're counting up the little water dots, let's say blue is the particles of water. In this side of the beaker, we have 18 molecules of water, and we have 6 molecules of solute. This is a 3-to-1 ratio of water to solute. On the other side of the beaker, if you counted up all of the dots here, we have got 18 dots of water, and we have 18 dots of solute. This is a 1-to-1 ratio between water and solute. That means that we have a 3 times greater concentration on this side. The concentration is not the number of molecules, it's the ratio between solute and solvent. We have a 3-to-1 solvent to solute concentration here. We cannot move the red particles across the membrane to even this out. So instead, we're going to move the blue particles across the membrane to even this out. So we're going to move water across the membrane. So we end up with, in this second picture, we have 12 water molecules, and we have 6 solute molecules. And then we have 36 water molecules on this side, and 18 solute molecules on this side. Now, if you're adding up these numbers, you'll see that we've created water molecules somewhere. Pretend we didn't create water molecules. This is just for the sake of the demonstration. But the solute molecules hasn't changed, but we've moved water until we end up with the same ratio. We have a 2-to-1 ratio. I said 2 to 2. This is a 2-to-1 ratio of water to solute. We have moved molecules across the membrane until we end up with the same concentration. The concentrations here are equal, even though the number of particles is not. Remember, concentration is the ratio. Let's do some practice with some diffusion and some osmosis. Okay. Diffusion, again, is the movement of solute. Osmosis will be the movement of water. You got to take a look at our concentrations. This is our intracellular fluid. We're going to more commonly use the word cytoplasm instead of intracellular fluid. Our cytoplasm and our extracellular fluid on this side. Inside of the cell, we have 5% salt. If we have 5% salt, that means that the rest of that percentage will be water. Means we have 95% water. Outside the cell, we have 10% salt, which means we have 90% water. Remember that things are always going to flow from a high concentration to a low concentration. The salt concentration is greater outside than inside. So the salt will go from out to in. The solute will move to the inside. Whereas the water, there's more water inside than there is outside the cell. So the water will flow outside. For our next row here, you see that the concentrations of the salt are the same. That means the concentrations of water are also going to be the same. Both have a 90% water concentration. If the concentrations are equal, it doesn't mean that nothing moves. It means that the rate of movement in and out is equal. So we're going to say that the solute will move in and out of the cell, and the solvent will also move in and out of the cell at an equal rate. Glucose or protein or salt are all just different types of solutes, so these will be the same. You just take a look at how much solvent do I have, 97% on the inside and 99% on the outside. That means that the glucose will flow from high to low. It will go from 3% to 1%. So the glucose is going to flow from the inside to the outside. So the glucose will go outside. And the water will flow inside. I'm going to skip to this last one here. For the last one here, they're telling us the concentration of water, not the concentration of solute. So if we have 13% water inside the cell, that means that we have 87% solute, whatever that solute is. And if we have 25% water outside the cell, that means we have 75% solute. So watch what concentration they're giving you. Solute's going to go from high to low. It's greater inside the cell than it is outside the cell. So the solute will go outside. And then the water is greater outside the cell than inside the cell. So the water will flow inside. You should notice a pattern here, with the exception of when the concentrations are equal, in which case you're going to flow both in and out of the cell. The solvent will flow the opposite direction of the solute. So water goes the opposite direction of the solute. Water goes the opposite direction of the solute. Okay, a little bit of a review. Diffusion. If you take a look here, we are focused on, these are our particles. We've got a higher concentration inside the cell than outside the cell. So we're going to go from the high concentration to the low concentration. The solute is going to leave the cell. We've got a higher concentration inside, and just put higher concentration inside, and we've got a low concentration outside. The opposite is true here. Here we have a higher concentration outside, and we have a low concentration inside. So we're going to flow into the cell. Now, here is osmosis. Here you care about the water. Water is the circles, and it's the only thing that can move across the dotted line. You're going to compare the ratio of the water to the solute. Okay, if you count up the water concentration on this side, 1, 2, 3, 4, 5 waters. We have 5 circles. And on this side, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. We have 15 circles. So this has got the higher concentration of water. We will move from a high concentration to a low concentration. On this side here, we've got way more circles than the other side, so we'll go from the higher concentration to the lower concentration. And it looks like we've got pretty much the same number of pentagons. Okay. Right here, this little simulation here, we have pure water on one side of a membrane, and then we have water and sodium ions on the other side of the membrane. This is a little bit beyond Science 10, but it helps explain what's happening here. The water molecules are going to associate with whatever solute is dissolved in them. So these water molecules that are associating with the solute can't actually cross over the membrane back to the pure water side, which means that they don't have the freedom to move across the membrane. The other water molecules do, so they're moving back and forth across the membrane, which ends up with more water being on the side that has the solute. So this side had a lower water concentration because of the solute until we moved water over. This is kind of osmosis at work. I think I'll save active transport for the next lesson, so this will be the last page for this lesson. Water moving in or out of the cell depends on something called the tonicity. The tonicity of solutes outside of the cell compared to the inside. The word tonicity basically is concentration. There are three types of solutions. We have hypertonic solutions, we have hypotonic solutions, and we have isotonic solutions. Now, these are describing the solution. This is describing the solution, which is going to be the extracellular environment. When we describe the tonicity of a solution, we're always comparing it to something. This solution is hypertonic to the cell. This solution is hypotonic to the cell. This solution is isotonic to the cell. This always involves a comparison to the cell. Take a look here. A hypertonic solution, hyper means a high concentration. Hyper means a high concentration of solute. Here, we're describing the solution or the extracellular environment. I'm going to draw a picture here where my blue dots are going to represent my water and my red dots are going to represent my solute. This circle on the inside is my cell. I'm going to start by putting water molecules. I'm going to put 8 water molecules outside the cell. And I'm going to put 8 water molecules inside the cell. Then I'm going to put some solute molecules. If this is a hypertonic solution, that means that I have a greater concentration in the solution outside the cell. So I'm going to put 2 solute molecules inside the cell, and I'm going to put more than 2. I just drew a bunch of dots, 8 outside the cell. This means that there's a greater solute concentration outside of the cell than inside. If you have a big solute concentration, that means you have a low water concentration. Water will move out of the cell. Water moves out of the cell because the water is more concentrated inside the cell. The water concentration is going to be opposite of the solute concentration. Water moves out of the cell. If water moves out of the cell, then the cell loses volume. This means that the cell will shrink. I have a picture here. This is blood cells in a hypertonic solution. If the solute can't equalize, then the water will leave the cells to go to the lower water concentration, which means that the cells kind of deflate. The water is leaving, like this. Hypotonic. Hypo, O rhymes with low. Hypotonic is a low concentration. I'm actually going to switch that to red still, because we're still describing solute, which we're keeping in red. This is a low concentration of solute. I'll keep the same colors where red is solute and blue is water. So I'm still going to have 8 water molecules on the outside. So that's 1, 2, 3, 4, 5, 6, 7, 8, and 8 on the inside. Now there's going to be a greater concentration of solute inside the cell. So I'm going to put, 6 dots inside the cell and only 2 outside. So I have more solute on the inside. We have a greater solute concentration inside of the cell. If we have a greater solute concentration inside the cell, that means we have a lower water concentration inside the cell. So the water moves into the cell because water is more concentrated outside the cell. If water moves into the cell, then the cell will expand. So if I go back to my picture here of these blood cells, the cells are expanding as water moves in. The threat here is that the cell might burst if too much water moves in, which is what's happening in this picture right here. Finally, we have isotonic. Iso means the same solute concentration. So an isotonic solution will have the same concentration. Once again, blue is water, red is solute. We're going to say that both the solution and the cell have the same volume, so the concentrations will be equal. If I put 8 dots of water on the outside and 8 dots of water on the inside, and then 4 molecules of solute in both, we have the same ratio of water to solute both inside and out of the cell. Solution and cell have the same concentrations, equal movement of water into and out of the cell. In and out of the cell, which means that the cell stays the same size. The key thing here is that the water is always moving. It's not that the water doesn't move if the concentrations are equal. Water is always moving. Here's a picture that's very similar to the picture that I just showed you on the other page, but for a hypertonic solution, water will leave, the cell will shrink. For an isotonic solution, the rate of water moving in and out is the same, so it stays the same. Hypotonic, more water goes in, and it will expand. This is a plant cell. I'm going to stop there, and in the next lesson, we'll discuss active transport. For this lesson, we discussed the 3 forms of passive transport, as well as the structure and function of cell membranes.